Okay, let's now turn our attention to things that are inside these epithelial cells, the so-called cytoskeletal elements that are found inside the cytoplasm of these cells. And there are going to be three kinds of cytoskeletal elements. They're going to be called microfilaments, intermediate filaments, and microtubules. Microfilaments, they're called filaments because they are filamentous, and micro because they are the smallest of the filaments, microfilaments. Microfilaments, regardless of what cell type you find them in, are made of actin proteins. Actin that has polymerized into filaments. Now, actin, before it polymerizes, is globular, so it's called G-actin. And when G-actin molecules polymerize to form filaments, we then call it F-actin. So, and that's how we form microfilaments, by the polymerization of G-actin into F-actin. This is an ongoing process that is energy dependent, so it's ATP dependent. So actin filaments are continually being polymerized, but they're not getting longer and longer and longer because at the same time that they're polymerizing, they are depolymerizing. So at one end of the filament, it's polymerizing. At the other end of the filament is depolymerizing. So we have this ongoing assembly and disassembly. So the F-actin molecules that form a filament at one moment in time will not be the same as the F-actin molecules forming that filament at a different moment in time because they'll have had ongoing assembly and disassembly going on in the interim. Okay, so at one end it's polymerizing, at the other end it's depolymerizing. That continual process is called treadmilling. Okay? So actin filaments are the filaments that we're accustomed to seeing in skeletal muscle. If you remember, in skeletal muscle, you have thick filaments and thin filaments, with the thin filaments being made up of actin, the thick filaments being made up of myosin and some other things. And so we can think of these actin filaments as being associated with contractile forces and with motility. And in fact, that's going to be true, but not just in muscle, but in other cell types too. So where we see microfilaments, we know that they're made of actin, regardless of what cell they're in, and we're going to anticipate that there's going to be some function that has either a contractile or a movement component to it. So, for example, earlier when we talked about cytokinesis, the division of cytoplasm, when a cell undergoes mitotic or meiotic division, and after nucleus divides, then the cytoplasm divides, there's a movement of the cytoplasmic elements into the two separate cells. And that movement is going to be facilitated by the presence of microfilaments. So the microfilaments are associated with the cytokinesis, the movement of cytoplasm during cell division. We're going to find microfilaments present in particular junctional complexes. We'll talk more about that when we look at those junctional complexes in a little while. So there'll be different junctional complexes that'll have microfilaments associated with it. And there are going to be microfilaments associated with certain surface modifications to some epithelial cells called microvilli. So what will form the core of a microvillus is going to be, again, microfilaments. So microvilli will contain microfilaments. Next, we have intermediate filaments. They're called intermediate because they are larger than microfilaments. They're the middle size of the cytoskeletal elements, so they're intermediate filaments. Again, they are filaments. But in contrast to microfilaments, we said all microfilaments are made up of actin. Intermediate filaments are made up of a variety of different proteins. Not all intermediate filaments are made up of the same proteins. Different cell types have different kinds of intermediate filaments in it, and we'll see that in the chart. It's not really terribly important for you to know the names of the proteins of all the different kinds of intermediate filaments. Just know that they are not actin. And the other thing that's different about them is that they are stable, in contrast to the microfilaments, where we said that they are continually turning over, continually polymerizing and depolymerizing. In contrast to that, intermediate filaments are stable. And the fact that they're stable is consistent with their function because their function is to provide structural stability to cells. Intermediate filaments are important in maintaining the shape of cells. In order for a whole variety of different cell types, in order for cells to perform their function, they have to maintain a certain shape. And so we want that shape to be stable, and so we want to have filaments in the cell that are stable, that will maintain that shape. So intermediate filaments are thicker than microfilaments. They are not made of actin. They're made of a variety of different proteins, and they are stable and provide structural stability to the cell to maintain cell shape. Okay, so listed here as different kinds of intermediate filaments in different cells. So we have type 1 keratin forming the intermediate filaments. We have desmin, vimentin, and so on. I'm not going to go through all of them. Different cell types have different kinds of intermediate filaments in them. But the common theme is they are stable and they are maintaining the structural stability of the cell. The third cytoskeletal element is microtubules. So they're called tubules, not filaments, because they are in fact hollow. They are hollow tubes. Hollow tubes. They are larger in diameter. We said microfilaments are about 7 nanometers. Intermediate filaments are about 10 nanometers in diameter. Microfilaments are much larger. They're about 25 nanometers in diameter. Like microfilaments, microtubules are continually assembling and disassembling. So therefore, they are also energy dependent in order to continuously assemble and disassemble. The function associated with microtubules are two main functions. One function is intracellular transport. Intracellular transport, the movement of things inside the cell from one place to the other, like the movement of vesicles within the cell. This is particularly obvious when you think about axons of nerve cells, where you have neurotransmitters being synthesized and packaged into vesicles in the cell body, but these neurotransmitter vesicles have to be carried all the way to the end of the axon to where the synapse is. And so we need to have a transport mechanism to move these vesicles from the cell body to the ends of the axons, and the mechanism that we will have will be microtubules. So that's just one example, certainly not the only example, where microtubules are going to be responsible for the transport of intracellular materials, often in vesicles. So it could be the movement of vesicles from the Golgi apparatus to the endoplasmic reticulum. We also see that in mitosis and meiosis, when we had movement of chromosomes during the division of the nucleus, the movement of the chromosomes was dependent upon microtubules. So microtubules form what are called the mitotic spindles during mitosis and meiosis for the movement of chromosomes. Okay. We said that microtubules are continually forming and polymerizing, and so it's energy dependent, and also the movement along microtubules are energy dependent. So microtubules will have associated with them what are called ATPase motor molecules responsible for this transport. And so, for example, in neurons, we will have two different motor molecules, one called kinesin for anterograde flow from the cell body to the axon terminal, dynein for retrograde transport from the axon terminal back to the cell body. But the point is that you have these ATPase motor molecules associated with microtubules. Now, the other major function of microtubules is to provide for motility. Of parts of cells. So, for example, cilia and flagella. The cilia, there are a variety of cell types that have cilia on them. The respiratory system has cilia on them, as an example, respiratory epithelium. And things like sperm have flagella, responsible for the motility of sperm. The movements of those cilia and flagella are dependent upon microtubules inside those structures. So inside a cilium or inside a flagellum, we will always find microtubules, and those microtubules will have sidearms of a motor molecule called dynein, and it's the movement of these microtubules one upon the other that provides for the movement of the cilium or the flagella. Because microtubules are continually assembling and disassembling, if we inhibit microtubule assembly, and colchicine is a drug that will inhibit microtubule assembly, and since we said microtubules are responsible for the mitotic spindles, the use of colchicine will interrupt mitotic spindles and therefore will inhibit mitosis. Okay, so colchicine is a drug that can inhibit mitosis by interfering with microtubule assembly. Cell adhesion molecules. Now, we've already talked about how epithelial cells have to be attached to surfaces, underlying surfaces of basement membrane and connective tissue, because they always have to be in contact with a source of their blood supply. And we've talked about how epithelial cells have to be in contact with neighboring epithelial cells. And so we need to have molecules that control this adhesion of epithelial cells to other surfaces. And so we're going to have cell adhesion molecules, which are responsible for the adhesion of cells to other surfaces. Okay? And there are a variety of different cell adhesion molecules, and some of them are calcium ion dependent, and some are not. I don't want to get into a lot of detail. I don't think that's important for us right now. Okay? But recognize some are calcium dependent, some are not. The point to be made is to think about the importance of cell adhesion molecules as being important for permitting cells to adhere to other surfaces, to other cells or surfaces, or to not adhere. So we want to have cell adhesion molecules to be able to be turned on or turned off. Cell adhesion molecules have to be able to turn on or turn off because, for example, during development, we have to have cells that migrate from one place to another as a part of development. Well, think about what's necessary in order for a cell to migrate. A cell has to first let go of the surface it's attached to, move someplace else, and then hold on to a new surface that it's attaching to. So we want to have cell adhesion molecules be able to turn off and then turn on again at another time. And so we're going to see cell adhesion molecules as an important component of embryonic development. And also, we need to think about cell adhesion molecules as being an important component in metastatic disease. Because in order for metastases to occur, a cell that's going to metastasize first has to let go of the surface that it's attached to, migrate someplace else, and then reattach to a new surface. So again, cell adhesion molecules are going to play an important role in metastatic disease. Okay, so now let's talk about the structural complexes that are involved in connecting cells to other cells. And these are called cell junctions or junctional complexes. And there are three categories of junctional complexes. We can call them barrier junctions, anchoring junctions, and gap junctions. Barrier junctions first. Remember we talked about paracellular transport, where material can pass from one side of an epithelial layer to the other side of the epithelial layer by passing between epithelial cells or around epithelial cells. Well, if they're going to be passing between epithelial cells, we want something available to us to control that passageway between the cells. And what we have that controls that passageway are called the barrier junctions. And there are two kinds of barrier junctions. They're called tight junctions, or zonular occludens, and zonular adherens. So they're being diagrammatically schematized here. So let me explain this diagram to you. This, what I'm outlining here, represents one epithelial cell. This is another epithelial cell. Here's another that would go off the screen, another epithelial cell. So here we're in the space between epithelial cells. Here we're between epithelial cells. Here we're between epithelial cells. Here is a surface modification to an epithelial cell, okay, that we'll talk about later. Here's the lumen. So here's the luminal surface that this epithelial cell is lining, and here's the basal surface of that cell. So if we want to get material from here to here without going through the cell, if we want to go paracellular, we want to pass through here or pass through here. And controlling that passageway, we have those tight junctions and zonular adherence, these barrier junctions. Okay? Those two kinds of junctions will always be seen together, with the tight junction of the zonular occludens being closer to the lumen and the zonular adherens being the one further from the lumen. They're going to be found side by side like that. And we'll take a look at what that looks like on an electron micrograph in a few moments. The major difference structurally between a zonular occludens and a zonular adherens is a zonular occludens is sort of a spot weld. It's a single point where these two cells are connected together. Okay? So if you go a little bit to one side or another of this, into or out of the screen, then that won't be there. In contrast, zonular adherens is a continuous ribbon of connection between cells. Okay, so it's a continuous connection between cells, whereas these are spot connections between cells. So zonular occludens are tight junctions and zonular adherens. So that's one category. That's the barrier, serving as a barrier to control passage of material in the paracellular pathway. Anchoring junctions, as the name implies, anchor or attach cells to other things, either attaching an epithelial cell to another epithelial cell, as is seen here, attaching this cell to this cell, or attaching an epithelial cell to a basement membrane, as is seen here. So the anchoring junctions that connect one epithelial cell to another are called desmosomes, where you have a thickening of this epithelial membrane and a thickening on this. And we're going to see that there are going to be intermediate filaments in the cytoplasm that are going to be particularly dense on both sides. So we'll see it on both epithelial cells. So that would make up a desmosome. Or if it's an epithelial cell connecting to a basement membrane, so you don't have another epithelial cell, then you only have half of a desmosome, just on the side that is on the epithelial cell. So you would call that a hemidesmosome. So desmosomes connect epithelial cells to epithelial cells. Hemidesmosomes connect epithelial cells to underlying connective tissue, basement membrane. And then the third category of junctional complex are the gap junctions. Gap junctions are really quite different from the others. Gap junctions, the function of gap junctions is to provide a pathway for communication between cells. It's basically a tunnel between cells, a place where we can have the movement of ions from one cell to another, and by moving ions from one cell to the other, we now have a pathway for communication between cells. So where cells need to communicate with adjacent cells in order to coordinate their activities, we're going to find gap junctions. So for example, when we get to the heart, cardiac muscle cells, in order for the cardiac muscle cells to coordinate their activities so that you have a coordinated contraction of these cells during cardiac contraction, we're going to want to have gap junctions to allow communication between cardiac muscle cells. If you look at the smooth muscle cells in the wall of the gut, again, you want the smooth muscle cells to coordinate their activity so you'll have a nice coordinated peristaltic contraction of muscle in the wall of the gut. So again, you'll expect to see gap junctions between intestinal smooth muscle. So gap junctions are really passages that allow for communication between cells. Okay, so what we've said is that tight junctions or zonular occludens are found close to the luminal surface. So here's the tight junction, close to the luminal surface, and it serves as a barrier between the lumen and the extracellular space. Okay? We're going to find microfilaments on the cytoplasmic surface of those tight junctions. Okay? There'll be actin or microfilaments in the cytoplasmic surface of tight junctions. Zonular adherens, which are always close by, just outside of or away from the lumen from the zonular occludens, zonular adherens are found here. Okay, and they also will serve as a barrier to movement between the cells, and they also will have actin filaments or microfilaments in the cytoplasmic surface of the cell adjacent to that complex. So we'll see that on an electron micrograph. Desmosomes, we said, are anchoring junctions. Okay, and there will always be intermediate filaments on the cytoplasmic side of desmosomes or hemidesmosomes. And because intermediate filaments are thicker than microfilaments, they're going to be more radiodense, so they're going to be much more apparent in electron micrographs. So we'll have intermediate filaments associated with desmosomes as well as hemidesmosomes. So let's take a look at this on an electron micrograph. So here is one epithelial cell. Here's another epithelial cell. Here's the lumen. So this is actually the epithelium lining the intestine. So this is the intestinal lumen. We see microvilli here that we'll talk about later. So here is the junction between two epithelial cells. Okay, right there we see the space between the two epithelial cells. So paracellular movement from the lumen to the basement to the connective tissue out here would have to run through here. And so that is guarded by these barrier junctions. So here we see a zonular occludens, and just below that we see a zonular adherens. We recognize these regions by the membrane densities associated with those complexes. Okay, so we see this thickening of the membrane, of the cell membrane, at those barrier junctions. Again, closer to the lumen, zonular occludens, further from the lumen, zonular adherens. Here we see an anchoring junction between these two epithelial cells. So since there's between two epithelial cells, it would be a desmosome, and we see the density of the intermediate filaments on the cytoplasmic side in both cells. Okay, so there we're looking at a desmosome, anchoring this cell to that cell. If we could look down here, it's off the screen, but if this epithelial cell is attached to an underlying basement membrane, then we would just see the thickening of the intermediate filaments in the epithelial cell, but not in the basement membrane, so you'd only see half of that, so you would call it a hemidesmosome. There are different cell adhesion molecules associated with desmosomes versus hemidesmosomes, different adhesion molecules. Because we have different adhesion molecules in desmosomes and hemidesmosomes, we have the possibility of different autoimmune diseases that affect desmosomes versus hemidesmosomes. So an autoimmune disease in which you develop autoantibodies against the desmosomal proteins that attach one epithelial cell to the other, that disease is called pemphigus vulgaris, in which it allows epithelial cells to separate from one another, forming more extracellular space between them and the accumulation of fluid or blisters between those epithelial cells. That would be pemphigus vulgaris. In contrast, bullous pemphigoid is also an autoimmune disease where you have autoantibodies, but it's against the proteins of hemidesmosomes, which are the junctional complexes that attach the epithelial cell to the underlying basement membrane. So again, you'll have a separation of the cell, but not from another epithelial cell, but from the basement membrane, but again, an increase of extracellular space and the accumulation of fluid and blistering associated with that. Finally, gap junctions, we said, are actually tunnels between two cells. So here's a schematic that shows us this. So here is one cell membrane. Here's the next cell membrane of the next cell. So here's the interior of one cell. Here's the interior of the next cell. This is extracellular in between them. And what we see is this communication, or this little tunnel connecting one cell to the next. So that is what a gap junction is. The proteins that make up the wall of that tunnel are called connexons. Okay? And it's within these gap junctions that we can have the passage of ions from one cell to the other. For example, calcium ions or other small molecules like cyclic AMP. And that is what allows for communication between this cell and that cell. Okay? And unlike other complexes where we had cytoskeletal elements associated with them, either microfilaments or intermediate filaments, gap junctions do not have any cytoskeletal elements associated with them. All right, now let's turn our attention to surface specializations. In other words, the epithelial cell, we said, is lining a surface, and that surface of the epithelial cell, that luminal surface, may have specializations on it to facilitate the function of that epithelial cell. So one kind of surface modification, surface specialization, might be microvilli. So that's what we're seeing on this diagram. These are each microvilli. They're prolongations or evaginations of the cell membrane. Okay, so prolongations of the cell membrane and occupying the space inside that microvillus, that's primarily filled with microfilaments. That's filled with microfilaments. So what we are accomplishing by having all these microvilli here is we have a tremendous increase in surface area of cell membrane, right? You know, if you compare that, if we didn't have those microvilli, then the entire surface would be that, right? But by adding all these microvilli, we've increased the surface area. So you would expect to see lots of microvilli on epithelial cells that are specialized for absorption, right? Because by increasing the amount of surface area, you've increased your ability to absorb. The more surface area you have, the more absorption you can do. So you would expect to see these specializations of microvilli on cells such as the epithelial cells lining the intestine or lining kidney tubules, both of which have lots of absorptive function to it. And again, as I said, occupying those microvilli are microfilaments. Another modification on the surface of cells are cilia. Here are cilia. There's a cilium. There's a cilium. Now, just for contrast, there's a microvillus. So let's look at the difference between a cilium and a microvillus. Firstly, cilium is much larger than a microvillus. But the other important difference is we said microvilli contain microfilaments. Cilia contain microtubules. Okay, so that's what we're seeing inside here. We're seeing microtubules. And remember, we said that the microtubules that are found in cilia and in flagella are there in order to provide motility to those structures. So these cilia are able to beat, to move back and forth because of these microtubules inside. So if we took a cross-section through a cilium, we would see this. And inside the cilium, we would see these microtubules. And the microtubules in a cilium or in a flagellum are always arranged the same way. We have this series of 9 couplets or doublets of microtubules surrounding a central pair of microtubules. So 9 doublets surrounding a central pair. That's the arrangement that you'll always see of microtubules in cilia and flagella. Coming off the sides of those microtubules, we have little side arms made of dynein. And remember, dynein is an ATPase motor molecule. And it's these dynein side arms that allow or cause these microtubules to slide over one another. And by sliding past one another, those microtubules are providing motility or movement to the cilium. So the fact that the cilium can beat back and forth is because these microtubules are sliding over one another because of those dynein side arms. Okay? And again, dynein is an ATPase motor molecule, which provides the motility to cilia and flagella. Okay? So here's a schematic version of that. Again, the doublets of microtubules surrounding a central pair. Okay? Now, we want to have these motile cilia in places where we are projecting into the lumen, in places where we want material in the lumen to be moved. So, for example, in the airway. In the airway, in the trachea and the bronchi, we're going to be secreting mucus into that lumen in order to coat the surface, because we want that mucus to trap foreign material that's coming into the airway in order to prevent or reduce the amount of that foreign material that gets all the way down to the alveoli in the lungs. So the mucus is going to be trapping that material. But we don't want to have that mucus just sitting there accumulating more and more foreign material. We want to be able to move that mucus out with its foreign material trapped in it. And so we want to move the mucus up along the airway. We want the mucus to be moved up away from the lungs, up the airway toward the pharynx, so it can then be swallowed down the pharynx into the esophagus. And so we're going to want these cilia to be beating to move the mucus up along the airway. And in order for those cilia to do that, we need the microtubules in the cilia. Similarly, we talked about the tail of the sperm being the flagellum, which is what allows the sperm to be motile. And in order for that flagellum to be motile, we need the microtubules with their dynein side arms to be functional to provide that motility. This all leads us to an understanding of a disease which is called primary ciliary dyskinesia. Primary ciliary dyskinesia, in which the cilia or flagella are not motile. And the reason they are not motile is because the dynein side arms are missing from these microtubules. So if there's an absence of the dynein side arms, then those microtubules will not provide motility, and therefore the cilia will not be motile, the sperm will not be motile. And therefore you would expect a male with that disease to be infertile, because his sperm will not be motile, and because the ability to move the mucus up along the airway is going to be impaired. You'd expect that person is going to have chronic respiratory infections. And that's in fact true with primary ciliary dyskinesia. Now there are, for reasons that we're not going to get into right now, there about half the time when a person has that, associated with that is also situs inversus, a reversal of the right-left symmetry, or asymmetry rather, in the body. And so when you have primary ciliary dyskinesia along with situs inversus, then it's called cardiogenic syndrome. So you can have this with or without situs inversus. If it's with situs inversus, it's called cardiogenic syndrome. Okay, so what we said we wanted to accomplish in this section was to describe the structure, function, and mechanisms of transport across epithelial cells. And remember, that can be either through the epithelial cell or around the epithelial cell. We wanted to talk about the different types of epithelial cells. Remember, we talked about the stratified or the simple, and then the different shapes, squamous, cuboidal, and columnar. And then we wanted to talk about the cytoskeletal elements, microfilaments, intermediate filaments, and microtubules, the cell adhesion molecules, the various types of cell junctions, and the cell surface specializations. So that's what we have accomplished.